Okay, thank you very much. It's a pleasure to be here. Sorry, there was a little bit of technical confusion here. We're just coming over. Actually, we've been at this meeting just on the other side of the river for the so-called LINCS project, which is this library, an NIH project called the Library of Induced Network Signatures. And I want to tell you a little bit about that first, and focus, say a little bit of what the goal of the project is, and then focus on what I think are some interesting visualization challenges. And it's interesting, coming right after Scooter, you're going to hear a lot of the same kind of things, and I'll show you some examples. I was a little concerned that when I get up here that I think every slide I have disobeys at least 2 of Bang's visualization rules, but I want you to see them as straw men set up for you to knock down and solve. So that's headed off those negative questions. Okay, so the goal of this project very broadly, and actually we heard a little bit of this also from Mike, is the notion is that part of what we're trying to do is to assess these intracellular signaling pathways in cells and how they function, and ultimately step up to tissues and networks between cells. And perturbation is, of course, one of the funnel ways to get at how these things function. I might say parenthetically, the very math of actually modeling perturbation, in my lab we do this using either regression methods or some imputed Bayesian inference methods. That in itself is sort of an interesting challenge because you actually have to code a significant part of the knowledge of how the perturbation is done into that. And it's something I won't touch on today, but if anyone's interested, we'd like to engage you in that. So the basic notion behind the LINCS project is that when a target here hits a perturbation in P, that you have a series of responses. One of them could be an intended response. And in the LINCS project that we're doing, and is also happening here at the Broad, so it's divided between 2 so-called data production centers. And we don't want the NIH to know, but we try to look at our own data. We don't just produce it. The aim is to look at therapeutic drugs. And one of the reasons for that, and I think it gets more and more true as you look closer into this, is even for efficacious drugs, we actually have really remarkable little understanding about why they work, and certainly the variation in response across different tissues and different patients. So one's trying to get at that sense of what is the initial response to that insult, what are those things that might be, in a sense, off target if this had therapeutic mechanism that would be toxic, and then what does the network do to respond? And what we've learned for a lot of these compounds, both in inflammatory disease and in cancer, is that often there are these paradoxical effects that are actually counter-therapeutic. So, for example, the inhibition of the AKT survival pathway itself directly leads to upregulation of pro-survival mechanisms. And so if we can begin to get at that, the notion is that one of the outcomes might be sort of a kind of rational approach to polypharmacy or combination therapy. So there are a lot of different types of variation. You can look at the effort here at the Broad, which you're going to hear more about from Bang, is really focused on large-scale transcriptional profiling, really excellent at looking at genomic variation. In our piece of this puzzle, we've been looking particularly at the level of proteins and at the level of environmental factors. So the overall goal here is to output some data that would be useful to the community in general, sort of a kind of large-scale perturbational study. We're, by training, biochemists and cell biologists, so you see that's the type of data we focused on. This tends to start with cell lines, but it's an explicit goal of the LINCS project to move into primary tissue. There's a certain amount of tool production here. I would say visualization is distinctly, and in my version of this, intentionally left off, because you will see that deficiency most definitely. And since part of our requirement from the NIH is access, the absence of effective visualization in itself is a challenge. So the kind of data that we've been collecting is a lot of single-cell imaging data, including dynamical data on cellular response to perturbation, a lot of multiplex biochemistry. You heard about RPPA assays in the talk just a moment. Here's an example of that up here. There's a lot of dose-response assay, and I'll show you a bit of that in a second. I mean, the simplest measure of what happens when you put some sort of a perturbagen or a drug on a cell is what happens over dose and time. There's a lot of cheminformatics, and as many of you may know, the visualization of chemical structure and similarity clustering is extremely sophisticated in pharma and almost non-existent outside. And those tools have been sort of inaccessible to us because licensing the best cheminformatics tool per lab is a couple of $100,000 a year. So, there's an area, there's an EMBL effort here, and I think that could be expanded. And then there's a lot on drug polypharmacy. So even the most selective compounds actually hit multiple branches Here I'm showing you a kinase against a kinase tree. I'll come back to it in a moment. And one thing we heard at this meeting from effort at Novartis is, in fact, the best SH and siRNA, in fact, has more polypharmacy in it than the average chemical compound. So there's a lot more of that going on than we sometimes imagine. One of the types of data that we've been particularly interested in is presenting image data on a large scale. This is sort of a screen here, and there actually is some software behind this. We use the software from the open microscopy environment that allows us to look at temporal dynamics. I'll show you an example in a moment. And so the informatic piece, the plumbing, is at this point rather nicely taken care of. But I think there's a lot of opportunity for thinking about how you visualize this sort of complexity in time and space and how you might put that onto a network depiction. Here now are the straw men for you to laugh at. One of the things we've had to do within our effort is to build out how do you take this complex data set and make it sort of accessible to the general community. And so we've drawn sort of ridiculous little pathways here. But what we're trying to overlay on this is a sense that for a whole series of compounds, we have a dose-response series. Because this was a snapshot a couple weeks ago, it's not filled in, but it is now filled in. So behind this are a couple of 100,000 data points collected by things like the Cancer's encyclopedia projects. Here's that polypharmacology that I showed you before. Right now, we actually get that through a commercial service. With Steve Gigi's lab, we're re-implementing that in mass spec. And with the commercial service, which is what pharmaceutical companies get, is that picture. Right? Now we get a big long list or we get the picture. And there is no visualization tool that I'm aware of to usefully take these circle sizes, which tell you how much inhibition, and overlay them here on this tree, which in itself, in its current version, is patented apparently. So therefore, reverse engineering it isn't going to happen in the commercial sector. You can then click around and you can find cell line information and stuff like that. And so there's a very large amount of this, and we're happy to, it's available on the web, I'll show you the URL in a moment, but we're really happy to engage anyone who would like to take that kind of a challenge that I gave you sort of a superficial look at and make it much better. So let me just tell you a little bit about this issue of dose response, because I think this illustrates this problem of multidimensional data. And so when you have a dose response relationship, typically it's a log linear plot. And the analysis to date has largely focused on the midpoint of that dose response curve, which you'd recognize as IC50 or GI50. And it's been presumed that that's the primary variable either for a single drug across different cell types or different patients, or alternatively for a different type of drug against a constant cell type. And we've recently done a multi-parametric analysis on this with a few thousand cell lines. And the notion, just to give you a sense of this, behind the cell line collection piece of the LINCS project is, no single cell line looks genetically like any tumor in the case of cancer, but that if you have a panel of these things, and this is an incredibly flattened projection of TCGA's cell line collection along with the primary genotypes seen in women with breast cancer, that you begin to map out at least a significant fraction of that genomic landscape. So this is unremarkable in genome space, so there are plenty of these things sequenced. But the idea that you would try to do biochemistry systematically across large collections of cell types is, I think, in its infancy, and that's really the thing that we're trying to emphasize. I'm only showing you dose response in this first version, but this is what you see across a cell line for a series of different compounds whose notional targets are shown there. And you quickly appreciate that, for example, this CDK4 inhibitor is sort of unique in actually giving you the canonical textbook dose response. And all these other ones, you have this variation on what's the maximum effect axis, or you see all these variabilities in slope. We tried to figure out actually, is there much of this variation? And you don't need to look at this picture in detail, drug on the bottom, target on the top, a couple of those parameters in the middle. The first one is the one textbooks tell you varies, IC50. This guy is maximum effect, this guy is slope. And you can see actually there's just as much variation, and mutual information tells you that actually that is either by drug type or it's by cell type. So the interesting thing that we focused on, and I'm trying to mainly get at visualization rather than science, but to give you a piece of this for a moment, is this variation in slope. Because conventional biochemistry gives us a great way to think about high slope. So high Hill coefficient is a consequence in the classical description of cooperativity. And in fact, you see it in these in proteasome inhibitors, which has 7 subunits and typically has a Hill slope. Even in patients, it's been shown of around 4. So what about shallow dose response? So here are a couple of shallow dose response curves. And here are a couple of steep response curves. And you can look at cell viability, and we've done single-cell imaging on this as well. And here you can begin to again see the kind of inadequacy of the depiction, because behind this dose response is a pretty large data set. But the interesting little thing that we teased out of it is where you see shallow dose response is in fact where, if you look at a single cell level, you're seeing maximum variance. So in fact, that's a consequence of a variation, which we actually think is not genetically determined in these cultures. So very slow dose response, maximum variation. You might begin to wonder how that actually works mechanistically, and this is sort of what lies underneath. Here's actually a depiction of cells that are treated now at about 0.2 times the clinical concentration with a drug that's used mainly in lung cancer. And what they're transfected with is a reporter for the activity of ERK. And we've taken them to a fractional response on that curve. This is a shallow dose response drug. And what you're beginning to see is what we think of as sort of variance in that previous depiction is probably some incredibly complex dynamic over time. So our group isn't bad at analyzing that dynamic with ARMA. We're pretty hopeless at developing a good visualization to allow other people to capture that type of depiction. Why might this be important? We're coming to the conclusion, if you could begin to drill down from between population and single-cell depictions of these kind of things, that you would realize fractional dose response is not that every cell is going halfway. It's that half the cells are going all the way and half the cells are going nowhere. And that's why probably for most classic cancer drugs, there's always a population that survives, and that actually regrows back up to have fractional sensitivity. And all of this is independent of genetic selection. That would be superimposed on this complexity. So I mentioned that before. So I want to show you one other type of experiment we've done, again, to highlight the visualization challenges, and then I'll wrap up with a little list of what I think are our most challenging problems. Here's a case where we measured a perturbational profile, much like we heard about in a previous talk, and then we're trying to predict a second thing. So measure biochemical state, try to predict drug response. And the data is sort of distinguished through pathways. It's done with, in fact, RPPA and other technologies. And these are the kinds of depictions we have. And what you're seeing here is sort of signal on the horizontal, perturbation on the horizontal axis, signal on the bottom axis, and then each of these is like a little individual box for a drug. And what we have used, I think this was the previous talk's example of stringing together the existing things with some lousy Python. So what we do is we've simply adopted heat maps, and thank goodness this isn't red to green, so I don't have to be totally embarrassed. And here's cell type on the top, and here's signal. But what this isn't giving you, and what the challenge I think here is, if you drill down a little bit, the data actually has this very interesting multidimensional feature. So depending on whether you slice on the drug axis, you slice on the perturbation axis, there are two different perturbations in this experiment, or you slice at the single cell level, each of them has a different information content. We actually now store these in HDF5 files, if anyone knows that very nice multidimensional matrix. We don't really know how to drill down and make these accessible for visualization. One little tidbit here that you could actually ask, is there any information in any of this kind of perturbational thing? Here's just an example where we took about a 70-dimensional protein assay, and we actually just plotted it, and we actually asked, how does this relate to drug resistance? This is a very important clinical compound in breast and other cancers called lapatinib. And what you can see is the protein state information is actually quite nicely predictive of sensitivity. And we've actually gone through and worked out what this is. I just want to skip ahead and say, if you actually look in this particular class of drugs, for which, by the way, there are 350 current active records in clinicaltrials.gov. This is the PI3 kinase mTOR AKT inhibitors. Mutational status is not, it really has no statistical power on itself. If you go through and you measure, this is still in cell lines, but there's some patient data coming along, you measure actually AKT or phosphorylation, so the notional targets. There's very little predictive information in that. What this multidimensional analysis has shown us is the primary information lives if you measure the levels of the upstream receptors. So that came out of a kind of statistical analysis of protein data, and it's distinctly different from what we're arriving at by looking at the genetic data. So the last slide till I get to the challenges here is to give you a sense that as we begin to develop visualizations for this kind of multidimensional perturbation protein space, we have some very early hints that there may be some very interesting canalization going on. So a wide diversity of genotypes seem to canalize into a much smaller set of biochemical states that seem to be predictive of drug response. Still preliminary, but I think that would be a hopeful thought from the point of view of visualization, because we're not always trying to show 10,000 dimensions to the fourth power. We're trying to zero down in on these causal networks. So, and a little bit on network mapping here. Here's a network map we've built of that, and I only want to highlight one thing, which is the lines here represent imputed connections, in this case by correlation, and the boxes represent the prior knowledge of the biochemistry. And here's another case where we have a challenging visualization because the information content is heterogeneous. It consists both of that which was supported by current data and that which is supported from prior knowledge. And almost all biochemical inference of this type actually has a significant prior knowledge component. So to just leave you with what I thought were sort of four interesting challenges. The one is kind of this multidimensional data. How do we move over time, space, single cell population, particularly when the matrices are sparse? Second thing is, how do we capture these spatial and temporal dynamics? We have a few sort of simple tools that microscopists have developed over time. I think those could be greatly improved. How do we look at this thing I skipped over at the end, but this relationship between imputed networks and prior knowledge networks? So it doesn't make a lot of sense in a biochemical network to spend 80% of your inference time figuring out if EGF binds to the EGF receptor. The simple fact is it does. So you need to put that in as a prior. We do that now more or less fairly routinely in the modeling, but we don't have a good way to capture that and then to see whether we're in fact sensitive to false assumption. And then the final thing is that a lot of this cheminformatic data, and if there was just one problem to come out for which we have, I think, the largest data set outside of the private sector, it is in fact that polypharmacy trees I showed you of kinase inhibitors hitting many compounds, where there's a mixture of evolutionary or structural relationship in kinases and then activity to be plotted on top of it. If you're interested in any of this, here's our website. You can email me. You can email Jeremy Mullick, who I chose because he's the only person with all three Tufte books living on top of his desk, and so he would be interested to hear to you about this topic. And these are fairly large projects of which I've given you only the slightest hint. But I appreciate the time, and I'd be happy to talk to anyone about it.
I'm going to tell you about the another links project that's housed here at the Broad. It's called the Connectivity Map. And we want to leverage the power of genomics to change the way we discover drugs. And in this project, we looked at two very different domains. One is disease, and the other one is chemistry. These are different domains where the people work in them are also very different. On one side, you have physicians, and on the other side, you have these sort of staunch chemists. And they can't interact because they speak very different languages. And what we wanted to do was to develop a common language that biologists and chemists could use so that these people can interact, and then would allow us to find connections between disease and drugs. And that language that we settled on is the gene expression profile of a cell. Every cell in your body has 20 to 25,000 genes. And the reason you have different cells is that these different cells express different subset of those genes. And just as different types of cells have a different gene expression profile, so does disease. And in normal cells and in disease cell, the gene expression profile changes, and that differential gene expression is the gene expression signature of that disease. It's essentially a barcode of that disease. And just as a disease has a barcode, so does a drug. So if you take cells in the lab and you treat it with a compound, you change that expression profile. So the idea is that you create this sort of common language, which is about which genes go up and which genes go down, and that allows you to look for connections by just looking at similarities in the expression profiles. And the CMAP, the connectivity map experiment, looks sort of something like this, where you take cells and you perturb it with a chemical compound at a certain concentration for a certain duration, and then you measure the gene expression from there. We also look at other perturbations like genetic perturbation, where we knock down a specific gene or use an overexpression construct where you overexpress a specific gene and then ask what happens to the other 19,999 genes and then measure the expression from there. And so what we get is fundamentally a big matrix of genes by experiments, 20,000 genes by 400,000 signature, which represents many more experiments because we do experiments in replicates and there's controls and so forth. And just to give you a sense of the scale of this project, it involves teams of people, and it cuts across many research sources at the Broad. And this take you through 10 cell lines of this Napoleon's March. You take 10 cell lines, 44,000 small molecules, 9,000 shRNA construct, 3,000 cDNAs. That generates 67 billion independent measurements that then get consolidated to 75,000 signatures. The reason it's 75,000 and not 400,000 is this map was made a year and a half ago. But so this is the data that we have, is 400,000 signatures by 20,000 genes. And what we do with this data is we ask, given any column here, how does it look, how does it relate to all the other columns in the database? And the way that we do that is we use a modified KS test. And it goes something like this, where if in the genes that are up in the query are also up in the target, and the genes that are down in the query are also down in the target, we call that a positive connection. And if the genes that are up in the query are down in the target, and the genes that are down in the query and up in the target, we call that a negative connection. And if the genes that are up in the query and also up in the target are down in the query and also up in the target, we said that doesn't make any sense. And so we call that a null connection, and we push that to 0. So essentially, if you do this across the whole database, you get a distribution of something that there's a big pile up at 0, and you have a distribution to +1 and another distribution to -1. There's two basic computational visualization challenges with exploring these sort of ranked lists. And the first is that we want to compare ranked lists. Right, so this is two query. For example, this is the query for gene A, and you might say that it looks a lot in gene expression space to overexpressing gene X. And it looks like the opposite of overexpressing gene Y. If you knock down gene B, it may look like a gene expression signature of drug W and the opposites and so forth. So we generate these ranked order lists, and we would like to be able to compare those lists. The other thing is we generate a list of genes that travel together, up regulators and down regulators, and we would like to be able to track these sets of genes across the data set. So this project is computationally heavy and requires a whole team of analysts that work on the data. But anytime that you generate statistical measures where the same number represents two realities in the data, I think there's room for visualization. This is Anscombe's quartet. And so there's 4 sets of numbers here that are statistically identical. They have the same mean on the X and the Y. They have the same linear regression and 16 other common metrics. So by statistic measures, they're exactly the same, but when you plot them, they immediately look different. And so that type of a problem also exists in our analysis having to do with the KS statistics. For example, the KS statistics can't tell the difference between a few genes that correlate very strongly versus many genes that correlate mildly. We might get the same connectivity score in that case. But the real question is, like, what do we do with the Anscombe's quartet of 500,000 column? Do we still try to plot it to get some overview? And so we've been experimenting with different processes of trying to get an overview of the data itself. And if you look at these ranked order lists, one simple approach is just to take the connectivity score of all the columns to a given query, and then plot that according to a scale from, say, -1 to 1, and then you get these sort of barcode plots. And this is essentially looking at a distribution where instead of using height, we're using opacity and density as a proxy for height. And one of the reasons for doing this is that it keeps, for me at least, it keeps the profiles as independent entities so that in the context of interactivity, you'll be able to select those signatures. And the other reason is that these conventional plotting of histograms are space inefficient. And so if you want to stack a bunch of these together, it's a little bit more difficult. And furthermore, these histograms are not so easy to compare on both axes at the same time. If you stack them vertically, you can see the lateral shift really well, but you can't, it's much more difficult to judge height. And if you stack them horizontally, now you can see height, you can compare heights better, but the lateral shift is more difficult to see. So I think these barcode plots give us a decent overview of the data and allows us to compare different ranked lists at the same time. But in reality, we probably won't look at the whole data set at once, and we'll just look at subsets of the data. So these plots, I think they scale reasonably to the amount of data we want to look at once. For example, this is 20,000 signatures, knockdown signatures, spread across 3 cell lines. So what you can see here is that you can see the distribution fairly well. You can sort of see the sort of center of mass shifting. And you can also judge the intensity or the number of signatures that are collected at the 2 extremes here, positive connectors and negative connectors. So we've been developing features to allow us to address those 2 fundamental challenges, namely to compare ranked order lists and also to track genes and signatures across different cellular contexts and different perturbation contexts. When you take your data and paint a very complex picture like this, I think there's a couple of approaches you can take. You can sort of learn to read these and trust that the emergent property from the data will paint a shape, give you a shape that you can learn to decode. So learning how to decode these complex barcodes. I think another approach is to take metadata from outside of the project to structure the data so that you get a sense of what you're seeing, to understand what you're seeing here. And we want to do both of that. So one of the things that you can do is select a number of signatures and say go out to KEGG or Go or MSigDB and then get a list of annotations back to see if these things are in fact in the same profile, in the same pathway or a different pathway. But I started with a list of numbers and got this far to visualization, so I don't want to do that and get a list back. But because lists of numbers, they tend to beget lists of numbers. And so I called Martin Wannberg and Fernando Villegas, who are just across the street at Google. They've together generated some very iconic representations. And this is Map of the Market that Martin made popular when he was at Smart Money. It's a way of showing hierarchical information where each branch of the tree is given a rectangle, and then it's tiled with smaller rectangles representing sub-branches of that tree. So, but instead of the map of the market, we were wondering if we could actually create a map of the cell using gene ontologies. Genes are connected to each other through pathways, and it may be possible to build a hierarchy and sort of develop a map of the cell. So if we were able to do that, then the exploration of this type of ranked list could be much more interesting, so that when you select series profiles, you can see that these are, in fact, part of the proteasome inhibitors. So now that these super rectangles, instead of representing sectors in the market, they represent well-curated pathways, right? And you can go along and look at this, and it could tell you that the majority of the connection is, in fact, translation inhibition, or the opposite ends are redox stressors. So the connectivity map is computationally and visualization intensive project. And so we've been developing these aspects of the tools to help us explore that space. And I think we hoped, my moderator standing up, so we hope that's between the poster session that Corey presented and my presentation here, we've given you a sense of what the connectivity map is trying to do, sort of connecting the genes that underlie diseases and the drugs that might be used to treat those diseases. All right, thanks for your attention.